All right, we're going to get started with the optical physiology or calcium imaging data here. Again, like we mentioned before, if this is not relevant to you, you're welcome to hack to code here or move to the other room for more quiet coding or debugging. Okay. So this tutorial is similar to the extracellular ePhys tutorial that Ben just went through, but all the data types are different because the data is different. Again, we recommend that for those on YouTube, check out the NWB file basics tutorial first, which covers the subject metadata, trials, and creating a file. So in this kind of brief tutorial, we'll cover how to create or represent an imaging plane in the NWB file, how to add your raw acquired 2-photon images or 1-photon images, how to add motion correction data, image segmentations, your ROIs, and then add your fluorescence and DF over F responses for each ROI. We start, again, just like the last tutorial, we'll do Python and then swap over to MATLAB in segments. So we'll start with our imports. You can just copy this to get all the data types that you need. And just like before, we'll create a base NWB file that has the required fields and then any optional fields that you'd like to add. Now, to start all of this, we have to create an imaging plane object, and that holds information about the area, the method that you use to collect your optical imaging data. So the imaging plane here has a number of fields that are required or optional that you can add, such as the indicator, the excitation lambda, and importantly, it has a device and an optical channel. And the device represents your microscope in this case, so store the name of your microscope, the manufacturer, and so forth. And the optical channel here allows you to store the optical channel, including a description of it and the emission lambda. And you can have multiple optical channels for each imaging plane. Now, to create these, you have to create the device in the optical channel first. You call NWB file .create device, and you pass in your keyword arguments, just like you've seen before. And optical channel, pass in your keyword arguments of description and emission lambda, just the same. Yeah. And then finally, you create your imaging plane by calling NWB file .create imaging plane. And you pass in all the parameters, including your device object and your optical channel. For more documentation on all of these different fields, you can click on here, imaging plane, to get to the documentation in PyNWB for the imaging plane. Okay, so now you have the metadata around the plane that you're imaging from. And the setup. Now let's get to the raw data. Let's say that you're working with 1-photon series data, and then next we'll cover 2-photon series data. For 1-photon series data, you have two options to provide the raw images to the NWB file. One is to pass all of it in as raw data, kind of read in from the file, read in from the images into memory, and then as like a NumPy array. So you would just call 1-photon series, give it a name, and the data would be your NumPy array for your time by X by Y of all of your images. Pass in your imaging plane, and then in this case, if you have a regular sampling rate, you can pass in the sampling rate here with the starting time of 0 or anything, or you can pass in your timestamps just like we've seen before for the time series objects. Alternatively, you can store your raw imaging data external to the file and keep it in .tiff format or usually .tiff format. And then within the file, you will store the paths, relative paths from the NWB file to your .tiffs. And that's provided here. Basically, you give it the dimension of your image objects, your images, and then you pass in a list of, a single or a list of file paths, relative file paths to your TIFF objects. And importantly, you have to say that format equals external to say that the data are not represented internally in the file. And since these one photon data are acquired or raw data, we're going to add them to the NWB file using add acquisition, just like we've seen before. And we're just going to add both of them in this example here. Now, 2-photon series data is very similar, but you call the 2-photon series class instead. Again, you pass in data, imaging plane, and you can set the rates or the timestamps accordingly. And you can store the data internally in the NWB file or externally as a set of relative file paths. And let's add those to the data to the NWB file as well. All right, let's pause and take a brief trip to MATLAB land. And just like before, we're going to create an NWB file, pass in the session description, identifier, session start time as keyword arguments, and we're going to create an imaging plane. Similar to Python, you have to create these objects first. So you call types.core.optical_channel to create your optical channel object, pass it a description and your emission lambda, and then you create a device for your microscope. This one is just naming it device, but you can pass it a device description and manufacturer. And then you create your imaging plane with the appropriate keyword arguments there. Now these are not linked to the NWB file just yet. Well, I guess the device is, but the other objects haven't been linked in yet. So you call NWB.general_optophysiology to set to add this imaging plane with this imaging plane name to the NWB file. And now that you've created that, you can create a 2-photon series object. It has to go in this order because the 2-photon series object references the imaging plane, so the imaging plane must exist first. And just like we've seen before, you call types.core. In this case, it's 2-photon series, but it's like the time series. And the fields are just the same, starting time, data unit. But now we have an imaging plane that is a soft link over to the imaging plane object that you already added to your file. Now this just creates a 2-photon series that's floating in memory, and next you want to add it to the NWB file by giving it a name 2-photon series and setting it to the object. In this case, the data are stored in the raw image data are stored internally. If we want to store the data externally as TIFFs, for example, then the keyword arguments are external file, and then you pass it a name or a list of TIFFs, relative file paths, and then actually that should be format external, and we set it to the object. Set the 2-photon series in the acquisition group of the NWB file. 1-photon series data are just the same, just swap out the data types. Okay, we can also store the results of any motion correction that you perform on the raw data by creating a image series object and a time series object. Now, your image series object here is the corrected forms of your raw data. You can name it, or you have to name it corrected in this case. And you pass it your raw data. These are typically stored internally, I think, but can be stored externally as .tiffs as well. And the formats should be familiar by now of, you can pass it a unit, a starting time, a sampling rate, and so forth. And then I think within our NWB support, the core schema right now, we support storing of XY translations. And so you would just pass an array with the XY translation from your raw to your corrected. And then you create a new object called corrected image stack, which holds your corrected data, your original 1 photon or 2 photon series data, and then the translation between them. And you can create a new motion correction object that takes in this image stack and organizes it into a type called motion correction. And because this is the results of processing of data, we're going to put that within a processing module named OPHYS. And so create a processing module named OPHYS on the NWB file, then add motion correction to that processing module. Next, you've collected your raw data, you've added it to the file, maybe you've done motion correction to it, and importantly, you want to extract your regions of interest and store those ROIs in the file as well. And to do that, we have a data type called plane segmentation. This is a subclass of dynamic table, very similar to how the electrodes table is a subclass of the dynamic table, and the units table is a subclass of dynamic table. In this case, each row represents a single region of interest, and there's a few required columns. There's you must have a image mask or a pixel mask, and you must link or reference to the imaging plane that you created earlier. And outside of the plane segmentation, once you've made that, you can put that into another object called image segmentation, and that can hold multiple of these plane segmentations in case that you perform different ROI extraction algorithms on the same images. Okay, so to do this in PyNWB, first, we'll just create an image segmentation object. It's empty, that's fine. And then we can call image segmentation.create plane segmentation and pass in the objects, the keyword arguments for a new segmentation of your data. Importantly, you pass in the imaging plane and the reference images that you are segmenting. But this will just create an empty table. To start adding your ROIs to the table, you have two primary options. One is to store these masks as full-on images. And in this case, each mask is the same size, the same dimensions as a single image frame from your data. So in this example here, we're going to create an image mask of, we're going to create 30 ROIs. Each one will be 100 by 100. We're going to pick a random patch of 5 by 5 to mark as 1, and then we'll add the ROI to the plane segmentation object that we created earlier. The values of this image mask can either be Boolean or continuous between 0 and 1. Alternatively, you can define ROIs using a pixel mask. This is a more sparse representation of these masks, where you store a triplet of x, y, and weight, and to define the mask. So we're going to do the same thing. We'll create 30 ROIs. We're going to pick random starting points in this example, and we will create a list of pixel masks that are pixel by pixel, and in this case, each one will have a weight of 1. And then you can build up your 5 by 5 or whatever shape array, whatever shape of mask you have, pixel by pixel. And you can add this to your plane segmentation object in just the same way. call at ROI, and then in this case, just call pixel mask equals your list of pixel-wise masks. If you have volumetric data, you can do something very similar, but you can call, they're not pixels anymore, so we call it, so it would be voxel mask equals your three-dimensional voxel mask. Okay, so once you've done that, you have a plane segmentation table with a bunch of segmented regions of interest, either using the image mask or the pixel mask or the voxel mask representation. Just like all the other tables, you can call a toDataFrame to print all of those out as a data frame object or as a pandas data frame and see it in a nice table form. Jumping to MATLAB, the workflow is a little similar, but it is similar but has a little more syntax to it, a little more code. So in this case, we're going to add ROIs, and we'll first generate a whole bunch of them. And you, just like in PyNWB, you have the options of storing image masks, pixel masks, or voxel masks. And but the dimensions are flipped because of how MATLAB and Python work. So in this case, the image mask will be y, x, and then n ROIs. And you will, is that right? Yes, okay. So we'll just pick a random starting point, and it'll be a 10 by 10 image, or 10 by 10 ROI as part of the full 100 by 100 image. And in this case, we'll just set the values to 1, the weights to 1. But it can be any Boolean or continuous between 0 and 1. Then we'll call it plane segmentation constructor. It's a table. It has columns, so we'll define these column names like image mask, and then we'll pass in the image mask as a vector data object, which represents a column, with the image mask data there. The imaging plane is a required field of plane segmentation, and you would link to it using a soft link as demonstrated here. Finally, this plane segmentation hasn't been linked to the NWB file in our MATLAB NWB file yet. So to do that, we first put it into an image segmentation kind of container and put the plane segment, put it inside there, and then add it to a processing module for OPHIS specifically, OPHIS data, by calling nwb.processing.set, give it the name of the processing module, OPHIS, and then your new module. Okay. Now, lastly, you have ROIs, you have raw images. You've extracted continuous fluorescence measurements from those ROIs, and you want to store those as well. So those are time series data, and we have a specific type for those kinds of data called ROI response series. The ROI response series extends time series, just like many of the other types that you've seen. The data array is n time in the first dimension and ROIs in the second dimension. And importantly, there's a dynamic table region object that references particular rows of a plane segmentation table. So let's say your data is 1,000 timestamps, time points, by 10 ROIs, then your ROIs object, your dynamic table region reference object, should have 10 elements that refer to which 10 rows of your plane segmentation table correspond to those data. In most cases, this you only have 10 ROIs, and you're describing the activity of each ROI in order, so this will just be 1 to n, but in some cases, your ROIs, you've scrapped some of them, and so there is no data for them, but you've segmented them. And so you can pick a selection, a subset of those data, to store in your ROI response series. Now, to create one of these ROI response series objects, you need to create that dynamic table region object to refer to your plane segmentation table. And we'll do that by calling plane segmentation.create ROI table region. And you pass in the row indices of the of which rows of the table you want to reference, or which ROIs you want to reference. In this case, it'll just be the first two. And then you can create your ROI response series object, passing in the keyword arguments for name, data, unit, and rate. And critically, the ROIs is your newly created region object above here. And to help any tools downstream know that this ROI response series object represents fluorescence data as opposed to some other type of data, we're going to add it to a fluorescence object and then add that fluorescence object into the same OPHYS processing module that we created earlier. We created that OPHYS processing module, and we would just call OPHYS processing module.add fluorescence, and the fluorescence is just an object that holds one or more ROI response series, which might represent different segmentations of your data. If you want to store DF over F data instead, just replace fluorescence here with a DF over F object. Finally, we'll write the file just like the same that I've demonstrated before and that Ben demonstrated in the ECEphys tutorial. Just call NWB HDF5IO with W as the mode and call io.write. And reading the data is just the same. In this context, we can read the 2 photon series data or any acquired data by calling read NWB.acquisition and passing the name of the object. And all of our plane segmentations and fluorescence data live within the OPHYS processing module, and you can print it from here. You can also use, if you're in a Jupyter notebook, you could just print read NWB file, and you get the interactive kind of clickable drop-down menus to navigate this. That's also an easy way, but if you want to do it in code, you have to use kind of a nested square bracket dictionary kind of notation. And just like in the other types of data, if you have a data set, those data arrays are read passively from the file or lazily from the file. So you're not loading a whole terabyte of data onto your computer at once. If you do want to load in all the data, you would just pass a colon and square brackets operator for your data at the end of the data field here. And if you want to slice and let's say pick the first 10 elements in time and 3 ROIs, you would slice just like in a NumPy array for passing a slice for each axis. So that's how you write and read the data in Python. And in MATLAB, storing the fluorescence is much the same. You have to create this dynamic table region object to connect your ROI response series object to particular rows of your plane segmentation or your ROIs table. In most cases, that's just all your ROIs. So you can use 0 to n ROIs minus 1. Because, as Ben mentioned in his tutorial, NWB uses 0 indexing. Your data field here must start at 0 to represent the first row. And then you can create your types.core ROI response series, passing in this new region object and the rest of the typical fields of data, the units, and the starting time. You would then put this ROI response series object within the fluorescence object to label it as fluorescence, and then add that fluorescence object to your OPHYS processing module. Finally, because we hadn't done it before, you have to add your OPHYS processing module to your NWB file. Then you can call NWB export to write it to disk with a given file name, and NWB read to read it from disk from the same file name. And just like in PyNWB, reading these data arrays is read passively or lazily, so you don't overload your RAM. But to read all the data, you would call data.load, and to slice the data, you would use typical MATLAB kind of slicing techniques, slicing syntax to read just a particular row or column or rectangular segment of the data array. Okay, so we covered, just to summarize, we covered creating the imaging plane and adding that to the NWB file, adding your raw data both internally within the file and externally as paths, relative paths, to the, let's say, external TIFF files. We covered how to store the results of motion correction and your ROIs from image segmentation, and we covered how to label, to store the responses of those ROIs over time. If you use Suite2P or Cayman or some of the other processing tools, you can use NeuroConv to read in the results of those processing tools, and NeuroConv will take care of all of this for you. You might have to provide some additional metadata when running NeuroConv. But if you want to customize or cover a more specific use case, you can go through this tutorial, kind of copy it and modify it as needed to store your specific data. Okay, so with that, do you have any questions on the optical physiology side of things? You notice it's very similar to the extracellular EPHIS, just the names of the objects are different, but the structures are very similar. Yeah. Multi-session registration. So just to be clear, you have multiple imaging sessions and you're registering them to each other or to some first, reference. Mm-hmm. Mm-hmm. That's a good question. So one way you could do that is in your plane segmentation table, you could track the global kind of ID for each cell as a custom column, and then that would be tracking it unique across the sessions. You could store the mapping as a separate object, but there's no built-in NWB object to store that at the moment. You can create a custom dynamic table, which is just any generic table, and put that information in there, but then you would want to put that into every file, probably. You could create it in one file and link to it from all the other ones, but that means that in order to read that data, both the file that has the mapping must exist while you're reading the other files. And so for some mapping, that's relatively small, I assume, of that mapping. And so I just recommend to put it in every file. But Cody, Ben, if you have any ideas on multi-session registration. Doesn't appear to be on. Okay.
With their structure identified counterpart, which is really different things. So the solution to that was that we provide per se on a custody column. They define, okay, for all your identified ROI, for all your ROI and ID, a given set, those map onto a global ID that is across different now, and in it, what I would probably recommend is on every custom column, on every table in NWB, there is a description. I would highly annotate that description, describing that process. So someone looking at the model the first time would be able to understand what it means, especially with a very different value of the process.
We should add that to the FAQ.
We've gotten it.
Yeah. Any other questions on how to store 2 photon data, 1 photon data? If you're working with fiber photometry data, we also have an extension in NWB for specific to that. But the workflow is very similar.